Hi, my name is Jason Young. I'm a writer for the Yale Scientific Magazine, and today I'll be interviewing Professor Jane Taylor and Jackie Becker. So to start off, how did you guys become interested in researching this topic?
My lab in the Division of Molecular Psychiatry for a number of years has been interested in understanding the neurobiology of addiction and alcohol disorders. And when Jackie joined the lab as a graduate student, we thought it would be really interesting to try and do some studies that were focused on looking at potential neurobiological basis for individual differences in vulnerability to alcohol use disorders using animal models.
Great. And so what was the sort of inspiration that led you to this discovery outlined in your paper?
The particular neurobiological, neuroanatomical targets we're looking at have been identified in risk for addiction-related behaviors before. In particular, NCAM, neural cell adhesion molecule, that the paper is focused on, is part of a gene cluster that has popped up repeatedly in genetic studies looking at alcoholism in humans. And so we decided to look in a model that was predictive of sort of inflexible addiction-related behavior to see if we had any differences in this target.
And what was insightful in terms of Jackie's approach was to take also a molecule that had been investigated in other forms of learning and memory function and dysfunction and try and apply it for the first time to an alcohol-based model. And what we were really excited about was that individual differences in the amount of this PSA-NCAM molecule in the brains of a outbred strain of animals that's going to have some variability, that actually predicted how much they would show altered patterns of drinking behaviors associated with being able to stop drinking, so-called extinction of the behavior, as well as reinstating drinking, which is sort of an animal model of relapse behavior.
And what is sort of, I think, particularly exciting about the work is that we were then able to go back and look not only at the pre-existing individual differences, but use a tool to manipulate the expression and actually show a causal relationship between levels of PSA and CAM and the ability to extinguish or quit alcohol seeking.
Great.
And that's something that we can really do with animal models that can never be done with human studies, because what would normally happen in a human study is you'd have some association between, for example, a genetic difference and a behavioral difference, but you can never go in and do the mechanistic study to really show that there's a causal relationship and not just a correlative relationship.
Okay. And I guess in general terms, could you guys describe the methodology used in this study, just sort of generally?
Sure. So the initial behavioral findings, we look at a behavior called Pavlovian to instrumental transfer. And so basically what this is looking at is the ability of a reward, in this case food paired cue. It's a tone that predicts delivery of food. The ability of that cue to drive later responding for the same food. And the idea is that the animals that are going to have increased responding during a cue presentation are going to have attributed more incentive motivation, or the cue is more incentivizing for them to behave. And so after screening animals on this food-motivated behavior, we can then look at responding for alcohols. We teach them to make a separate response, nose poke for 10% ethanol. And after training, we can then extinguish the behavior, which basically means we put them in the same boxes and they make the same response, but they don't get a reinforcer and they don't get the presentation of the cues. And then we can do cue-induced reinstatement where we play those cues, so a flashing house light and a white noise that had previously been paired with alcohol, and look at relapse or reinstatement of the response.
Great. And so to go back to the sort of results of the study, how is this discovery significant, and what are some possible maybe clinical applications for this discovery?
Well, I think at this stage, it's really not possible to have any direct clinical applications. It might indeed be possible to see whether in humans, people who have polymorphisms in genes associated with NCAM, whether they might be more at risk for developing some alcohol use disorders. And certainly you could look at the ability of cues to invigorate their behaviors in a non-invasive sense. I think what it really allows us to begin to do is to try and find some genes and molecules that might put people at risk. And obviously, if you knew you had those sorts of risks, one might be more careful about engaging in extensive drinking behaviors in the future.
Right. And so for your lab, will this project lead to any further studies? And if so, what are they?
The project has already led to many, many studies. Jackie's got 2 papers that she's about to submit. We're really excited about both of them, and I'll let her maybe summarize a couple of them.
So, in addition to this cumulative behaviors, there's a couple others we're interested in, and one is just Pavlovian approach. So it's there's no response component. The animal doesn't have to perform an action to ever get a reinforcer, and we've looked at individual differences in Pavlovian approach, which is just the ability of a cue to motivate moving to a magazine in our task, and how that relates to a different form of inflexible alcohol seeking, which is habitual and compulsive like ethanol seeking. And so habits are behaviors that occur despite changes in the value of their outcome, or despite changes in the relationship between the behavior and the outcome. And compulsive-like behaviors are behaviors that occur despite adverse consequences, so when punishment or negative outcomes are introduced. And so we're looking at some epigenetic targets and seeing that in brain regions that are related to those behaviors, we do see epigenetic differences in serotonin receptors. And I'm also working on looking at the role of this brain region in its paper, the infralimbic prefrontal cortex, and habitual behavior in general, and looking at manipulation of dopamine signaling and how that can modulate the expression of habitual and goal-directed behavior. And also moving forward, we're looking at sex differences in some of these behaviors and the predictive validity of approach and PIT of alcoholism-related behaviors. And we've seen that there are, in fact, sex differences in the ability to predict risk, which I think is particularly important, not necessarily as a direct pharmaceutical kind of thing, but in general in society when most drugs and therapies are tested on men and not women, it's important to remember that sex differences are very real.
So in general, a lot of the research is really trying to understand how good decision-making skills and brain plasticity is important for normal behaviors. And what happens in addiction and alcoholism is that there are changes in the brain, and that one can have pre-existing risks for some of these changes to be exacerbated by exposure from the drugs of abuse. But if you think, as Jackie mentioned, one of the things we're interested in is habitual behavior. That's what you think of as is the problem in addiction, that you can't break the habit and you compulsively seek the drug. So those are the sort of psychological processes which are part of the psychiatric definitions of addiction that we're trying to understand on the molecular and cellular level.